Yeah, so thanks everybody for being here. My name's Paul Steen, and I work with the Huron River Watershed Council. MiCorps is this really neat organization because it's this combination of state government and educators at Michigan State, and we have nonprofits that are also involved on the leadership team, like my organization and the Michigan Lakes and Streams organization. So this interesting combination of people, and I'm happy to help out on the Lakes program. Though the majority of my work is done with stream monitoring, so I handle a lot of the stream monitoring side of MiCorps. But for Chlorophyll-a, Joe and Eric dust me off and get me in front of the camera so they can have a break. And I'm happy to, because I've been doing this for a long time, and happy to kind of get back in the lake space and help you all with understanding chlorophyll-a, which is probably the most technical of our parameters that we measure. So you've probably seen by now that year one, if this is your first year in the CLMP, you can't register for chlorophyll. We ask people to wait for their second year and beyond until they can register. And that's because it's just a lot of procedure, it's a lot of numbers to know. So we kind of like to ease you in slowly into this parameter. So if you're not registered this year and you're just here to learn, that is great. My hope is to get you excited about it so that next year you'll be ready to go with it. So in this training, we're going to talk about what is chlorophyll. We're going to talk about when to sample. We're talking about how to sample. And then when sampling goes wrong, when things come up that ends up with your samples getting rejected, we want to make sure you know how that happens so you can prevent it for your samples. So what is chlorophyll? Well, go back to your high school biology days and you'll know that chlorophyll is the pigment in green plants that turns the sunlight into food. Green plants includes algae. And of course, lakes are full of algae, and lakes need to be full of algae. That is the base of the food chain. It provides all the energy from the sun going up into fish, into frogs, and eventually birds, bigger fish. So all the way up the food chain. If you didn't have chlorophyll in your lake, you would have a pool. And if you wanted a huge pool, go ahead, but our lakes are special because they do have all these living things in them. Too much chlorophyll can be a bad thing. So if you look at the picture on the right, that belongs to Ford Lake, which is pretty close to my house. And Ford Lake has too much phosphorus, and August every year it has these big algae blooms. All the algae kind of comes up at once, creates this sludge on the water surface. Eventually all that algae dies, sinks to the bottom, takes up dissolved oxygen. So just not a healthy lake. Hopefully your lake is more like one on the left, where there's going to be algae in there. You can see people out boating and fishing. Without chlorophyll, we wouldn't have fish in our lakes. So it's a very important thing to have, of course. And when it comes to predicting the trophic status index, we consider chlorophyll the best predictor. So if you were here earlier in the day to hear from Eric about the trophic status index, he taught you that is the amount of productivity in the lake, or the amount of energy produced in a lake. Chlorophyll-a is the most direct measure of this because it's directly measuring the amount of production of energy through the algae. Secchi disk does get at this, but Secchi disk can be confused by sediment issues and light issues. Phosphorus does this as well, but phosphorus is really a measurement of how much algae can be produced, which is then a measurement of the amount of productivity. So chlorophyll essentially is a more direct link to that. So It's great to do all three of those parameters. It provides, we don't want you to say just do chlorophyll, because I think one of the main reasons is because there are places in the chlorophyll measurements where you can make mistakes. And so we still want you to get Secchi and we still want you to get phosphorus in case something bad goes with your chlorophyll measurements. And plus those two measurements are very easy and quick. So you should always continue to do those as well as chlorophyll. So, like I said, it's a complicated system. So let's start going through what do you need to know when it comes to measuring chlorophyll. Well, the first thing is you just need to know when we want you to sample. So here is just a screenshot of what our chlorophyll sample schedule looks like. And if you register for the program, you will get one of these in a printed version. They're also always available digitally. Here's how you get to it on the top there. On our website. And now you'll notice on the left-hand column, we have all the counties listed. So you find your county there. The column after that is where you eventually will drop the sample off. So we'll get back to that at the end of the presentation. And then the third column are those sampling dates. So check this out. There's 5 sample dates. The first 4 always happen between the 10th and the 20th of each month. May, June, July, August. Will always, that sample always is in that window. So you want to make sure you hit that window when you're sampling. And then you'll see that 5th sample is a strange date that doesn't seem to follow any sort of rhyme or reason. That 5th sample is the same time you take your phosphorus, your summer phosphorus sample. So that's why we did it that way, so that you can just go out that one time and get that final end of the year sample. In southern Michigan, that will be getting kind of close to the end of September, while in the UP, you'll be taking this at the end of August. So if you're in the UP, you'll have sample 4, which happens August 10th through 20th, and then a week or 2 later, you'll be taking that sample 5. So that's how we distribute when those samples are taken. You'll notice there's also 2 turn-in dates. Samples 1 and 2 get turned in on June 24, and that is also the same day you turn in your spring phosphorus samples. And then samples 3, 4, and 5 get turned in the same time you turn in your summer phosphorus sample. So that's all done, so you only have to make, all told, 2 trips to that drop-off point. And it also gets the lab moving, right? We're going to give this stuff to the Eagle lab. They're going to start processing. So that's why we wanted to get some initial samples in as quick as we can and earlier in the year. But because we have 2 turn-in points, that means we need 2 data forms. So that's why when you get your packet of stuff, you'll see 2 data forms. And data form 1 is what you fill out for May and June, and data form 2 is what you fill out for July, August, and then the sample 5. It's very important that you collect at least 4 of those 5 samples. 5 obviously is optimal, but we need you to get at least 4. If you only get 1, 2, or 3, then you're not collecting enough data to properly understand what's happening on your lake over the course of the summer. And that's because your lake will change based on a variety of conditions. Things that happen on a lake. Here's a graph that shows how lakes change. Well, the algae population in your lake will change. In the spring, after turnover, there's usually a lot of sediment turned up in a lake. And with a lot of sediment comes a lot of silica. And diatoms are an algal group that needs silica. So when you have silica, you have a bloom of diatoms often. And then summer progresses, those diatoms will drop off, and summer, July, probably June, July is like the peak of green algae. As the summer progresses and the water starts getting warmer and warmer and warmer, that's when your cyanobacteria or your blue-green algae start coming in abundance. For example, the picture I showed you of Ford Lake, it's cyanobacteria. It's that really thick, sludgy algae. Come fall, you'll have a turnover again, which means more diatoms. So the whole point of this slide is just to remind you that lakes change over the course of a summer. We can't just do all our sampling at one point and call it good. We sample over the course of the whole summer, so we're picking up on these different things that are going on. Okay, so let's talk about some of the equipment. When you register with our program, you will get some equipment in the mail or you pick it up at our in-person training. And it's a lot of different stuff. And I'm not going to go through all of these, but I wanted to show you some of the most important ones or some of the pieces that maybe are a little tricky. And to do that, I like to actually get full screen on here. So I'm going to stop sharing so that you can see me bigger. And I'm just going to show you some of this stuff that you'll get in the mail. So this piece is called your depth integrated sampler. It's meant to be disassembled. All right. And that's because this will be dropped in the lake, it will get wet, potentially it will rust. So every time you disassemble it and you just dry it off, leave it in your drying rack in your kitchen sink, and that will do the trick nicely. So the way you take this apart, this top piece unscrews, and there's a rubber stopper right here. The rubber stopper's got 2 holes in it, 2 tubes. You'll eventually, as you start using this, you'll see, water is going in one of these tubes and air is coming out the other. So that's how your depth integrated sampler fills up. Take that off just for now. There's a little chain on here. This is what keeps the whole device from just floating away. So when you're disassembling it, you can just get that out of the way, and this will come out. And you'll need to do that often because you will be rinsing this with lake water. Now, in your sampler itself, there are a couple of loose weights, and that is to prevent, well, when you drop this thing in the water, you just want this really quick, rapid descent down. So the weights, help make that happen. So you have a nice drop. But notice they're loose. They're not glued in, because if we glued them in, we would never get this thing properly dried. So these are sitting in here loose, and you have to be careful you don't drop them overboard, because that happens. All right, so I'm sure my sound is picking that up really nicely. Now, one thing that you will have to provide is a rope to attach to this sampler. So just even a clothesline will do. And you mark this rope every foot. You see how you don't have to put like a number on the rope, but you do need to mark it every foot so that you can, when you start using this, count off the number of feet that you're letting out. All right, I'll move that to the side and just show you a couple other of our trickier pieces of equipment here. This syringe will be needed later on for our filtering process. The tip of the syringe has threads. However, the things that we're asking you to put on the syringe do not have threads. So here's a piece of Tygon tubing that you will be using for the process, and this does not have any threads in it. So until you get used to it, it might be a little tricky. You'll be asked to put the Tygon tubing onto the syringe. And the way you do that is you apply pressure and you twist. And, it doesn't fit perfect, but it's pretty tight. You can get a nice tight seal on that if you just kind of twist and apply pressure. And that's, you'll see in the video later, that's how that works. And then we have another similar piece of equipment. This will eventually hold your filter. And once again, it does not have threads on it. But again, you can just twist and apply pressure. And you can get this thing in here very well without it any water leaking out of the edges. So this is called the filter holder, and it unscrews. And in the filter holder, there'll be a little ring. So this is needed to prevent leaks. So be careful that you don't lose this. It may be kind of a clearish circle, or it may be brown like that. So just be warned that that's in there, and you don't want to lose it. Now, if any point during the sample season you do lose pieces, the bottom rusts out of your sampler, you drop your weights overboard, all this stuff happens, and it's okay to get in contact with Jean Roth, who's the person sending you all this stuff, and she will get replacement equipment to you. All right, so that's, now you don't need to see me big anymore, so I'll go back to my PowerPoint presentation here. All right, and we will pick it up again. Okay. Here's the sampler. I just showed you a different view of it. If you have done this before, you don't need the big sampler. You don't necessarily need all the pieces I just showed you, but you do need every year the stuff that gets used up, the consumables. So that will include filters, that will include vials that you eventually put the filters in, your magnesium carbonate dropper, we'll talk about all this stuff, Ziploc bags, labels. Of course, you're going to use up that stuff every year and turn it in, so you'll always need a fresh supply. So when you register for your lake, you'll see the option to buy a full kit or a resupply kit. So that's what that's about. When you get your filters, you'll see they come in a baggie that looks like this, and it'll have a label on the outside that says, use the filters and discard the packaging. So all of those blue little pieces in there are essentially junk. You can throw those away. They're in there to prevent all the white filters from sticking together. These white filters will do that. So, but you definitely do not want to confuse the two. Oftentimes people think these blue separator sheets are the filters because it looks more like what you would put in your coffee machine. It's kind of translucent, but it's very plasticky. And it's not a filter. You can't push water through it. The white filter, a lot of if you're just going by your instincts, you would think this isn't a filter because it's this opaque, probably even more so plasticky. It just doesn't look like a filter, but that's because the pores in these things are so fine that it just doesn't look like how you would normally use a filter. But as far as lab use goes, it's just a really good small pored filter. So if you use the blue stuff on your sample, we'll have to reject that sample, and it happens to multiple people every year. All right, so just remember, blue is bad. It just says right there on your baggie to discard those blue pieces. Please do so. Please don't use them. There's things that you're going to provide. I talked about the line that will go onto your sampler. You'll also need a variety of other things that will come up over the course of this training. Here they're listed here, and we'll talk about them as they come up. Okay. So there's two main parts to carrying out this process. And first you have to get the water from the lake, which requires a certain process. And then you have to filter the water from the lake, which requires a second. So first we're going to talk about getting the water from the lake, which we call a depth integrated water sampler or water sample. So a lot like our other parameters, obviously you need to head out on your lake. It's important that you don't just throw your anchor in and cause a big plume of sediment that you have to sample into. So it's a better practice if there's no wind, you can just cut the motor and kind of drift over that sample point. And then if it's windy and you have to anchor, it's better to anchor upwind and let the wind then push you into that sample point. So ultimately what you're just trying to do is keep the water as still as possible. Start filling out your data sheet on the way. And then you'll do a Secchi disc measurement. You'll probably be enrolled in Secchi, so you'd have to do this anyway. But the Secchi disc is an important part of the chlorophyll procedure. We'll talk about why. So just as a reminder of a Secchi disc measurement, you slowly lower it until it disappears from view. You raise it again until you can see it. And then the depth you record is the average of those two depths. A reminder not to use sunglasses. Do it in the shady side of the boat if there is shade out there for you. Yeah, I guess that's all I have to say about that. So that's step one is getting the Secchi disc measurement. Why do we need Secchi? Okay, so there's a little cartoon here so you can kind of see what's going on with the sunlight on your lake. So our eyes let us see down to a certain depth. That's the Secchi disc depth. That's the point. And depending on the water clarity, that's the point in which our eyes can see. However, light penetrates further than what our eyes can see. And if light is penetrating down further, that's also where there's going to be algae growing. So now studies have shown that twice the Secchi depth is very close to how far light can penetrate to the extent that it can cause algae to grow. Since we're interested in sampling the entire algae community, both at the top of the lake, in the middle of the lake, closer to the bottom of the lake, we want to get water that has all of that algae in it. So we need to drop our chlorophyll sampler to twice the Secchi depth. And what we do is we're going to slowly pull this sampler up so that as we pull it up, it slowly fills with water. That's why we don't just stick our sampler right in and grab the top, we just have this tiny little tube that lets the water in. So as you pull it up, it just slowly takes water from all those different depths. On your data sheet, there's a spot for Secchi depth. And then right next to it, there is a spot for the composite sample depth. And that would be twice the Secchi. So you just multiply it by 2. Now, some of you, I'm sure, are asking, but my lake is too shallow. I can't go twice the Secchi depth. My Secchi is 10 feet, but my lake is only 15 feet. Well, if that's the case, what you do is just go as deep as you can. You never want to sample at the very, very bottom of the lake because we don't want to hit the bottom of the lake and plumes of sediment come up. So in that case, you always measure you get the sampler down to about within a foot of the bottom of your lake. All right, so that's how you handle those situations. Totally normal for that to happen. If your lake is only 15, 20 feet and your Secchi's 15, that whole lake has algae from the bottom to the top. So then you want to sample that whole area. Okay. So now we've done the Secchi. We figured out our depth that we're going to drop our sampler as well. So you take the sampler apart. As I showed earlier, you take off the top, you take off the stopper, you take off the chain. So you just have this. So give this a couple of rinses just to have normal lake water in there and not your sink water from earlier when you cleaned it out. You reassemble your sampler. Okay, and now you'll grab the clothespin that we included in your kit, and you'll grab your rope. And if you take your clothespin and you mark your rope at that composite sample depth. So let's say our composite sample depth is 20. Then we're going to count out on my hashes down to 20 so that we have 20 feet of rope. And then we're going to mark it with the clothespin. So now we know exactly what 20 feet of rope is. And we're going to have this big just pile of rope. What you do with this is just put it in the water so that it's ready to go. And then you hold your sampler nicely over the water surface and just drop it. It will fall quickly. It will pull the rope down with it, of course. And then you want to stop it when your clothespin is at the water surface. So you know that your sampler is exactly at, in this case, 20 feet down. Just let it free fall. Once the sampler uses up all the slack on your rope and you know it's at the proper depth, you start pulling it up. So just a steady hand over hand action at a steady rate. The reason a steady rate is important is because we care about the water at the bottom, and we care about the water in the middle, and we care about the water in the top, and we want to make sure all of that water is going through our stopper and getting in the sample. Once you pull it out, you have to check how much water you actually have in here. So disassemble it enough that you can see how much water we have. Then you want to get it at least halfway full. What you don't want to do is have it filled up to the top. Because if it's filled up to the top, then you don't know, maybe I pulled too slow and this thing filled up at the bottom and I don't even have water from the top of the lake. But if there's some emptiness in here, then you know that it's sampled from the bottom to the top. However, if you only have a little bit of water, you probably don't have enough to actually analyze the sample. So you need about half. Half is good. You know, as long as it's half and above, but not full, you have enough. And if you didn't do that right, you have to repeat the process again, and you'll have to change the speed at which you pulled in your sampler. So that takes everybody some practice. No one, I'm sure someone out there gets it right on their first try, but lots of people take multiple attempts to get this right. And then once you do it for a few tries, you'll have a better sense for the next time you sample about how fast you need to pull it up. So it's fine if it just takes you a little bit to get that right. In your kit, you're going to have some smaller bottles that look like this. So what you do is, now you have this separate. This is your lake water, and this is empty. And so you want to swirl this gently, because you're trying not to break apart all the algae cells in here. So you give this some swirls, and just a little bit to rinse and then dump that. Rinse, dump. So now this is nice and clean. And then fill this up to the top. All right. You'll actually have two of these. Yeah. You'll have two of these, and you want to fill both of them up. All right. So that's your ultimate goal here. Whenever we mix up the chlorophyll, we're always doing it gentle. You could also do this, but you never do that. It's just slow and easy, swirly mixing. What you are allowed to shake up like crazy is this little bottle of magnesium carbonate that we give you. So you do shake that up crazy, and then in each of these smaller amber bottles, you put 5 drops. And what that does is it helps stabilize your algae cells, just prevents some degradation, and makes them just hold up a little bit better through the next steps. And then you take both of your bottles and you put it in your cooler, and which you should have ice with you. You're also putting like your phosphorus samples in your cooler, for example, and you're done. You bring that back to shore. Okay, so that's the part I can show you with still pictures and through video. But the next step involves taking your amber bottles and going through this procedure to put your algae cells onto filter paper. So that requires better close-ups. And to take care of this issue, we have a YouTube video, and I recommend that you maybe just take your phone or something and snag a picture of this YouTube video. I'm sure Eric also has this listed in other places, like online, on the MiCorps.net, so you can find it. But I recommend the first time you do this, you watch the video very carefully, and then maybe give it a quick perusal every other time you're about to do the sampling, just so you remember all the steps involved. Actually, I'm sure you probably put in chat just now that link so that people will have it available for them. All right, so let's go to YouTube. And we're going to watch this video from Tamara.
With the Michigan Department of Environment, Great Lakes, and Energy. Today we're going to demonstrate how to filter a chlorophyll sample using the MiCorps Cooperative Lakes Monitoring Program protocol. To see how to collect a chlorophyll sample, please see our other video on chlorophyll sampling. After a chlorophyll sample is taken, the brown bottles should be kept in a cooler with an ice pack out of direct light. Before you take your bottles out of your cooler, check and make sure you have all of your equipment. A coffee filter or paper towel, a 60 milliliter plastic syringe, flexible plastic tube, filter holder, 2 membrane filter discs, tweezers and a large safety pin, 2 sample storage vials and caps, 2 chlorophyll sample labels, aluminum foil, and a fine tip permanent black marker. The best place to filter a chlorophyll sample is at a sink inside out of direct sunlight. So let's get started. Unscrew and open the filter holder. Using your tweezers, remove the black O-ring and set it to the side. Open the bag of filters and note that there are both white and blue circles in the bag. The blue pieces are the filter separators and should not be used. The white pieces are the actual filters. Using your tweezers, remove a filter without touching them with your fingers. Center the filter on the screen side of the filter holder and place the O-ring on top of the filter. Screw the filter holder back together until it is snug and place it to the side. Grab the syringe and the small piece of clear plastic tubing. Push the tubing onto the syringe. It does not snap in place but will fit snugly. Set the syringe down and swirl or invert one of the brown bottles with your chlorophyll sample to make sure nothing has settled to the bottom. Next, we need to rinse the syringe with the sample. To do this, use the syringe and tube to draw up enough of the sample to fill the syringe and then empty it into the sink. Immediately use your syringe and tube to collect another sample from the bottle. You will need to reach the 60 milliliter line. With the syringe pointing up, tap it to force air bubbles to the top. Over the sink, push the syringe plunger until the end of the plunger is lined up with the 50 milliliter mark. Remove the plastic tube and attach the filter holder to the syringe. Like the tubing, it does not snap into place, but you can get it to fit snugly. Point it down towards the sink. Slowly push the sample water through the filter holder. The pressure should be such that you see a rapid drip. It should not be a steady stream of water. Pushing too hard may rip the filter, so avoid excessive pressure. As you push the plunger, make sure that no water is leaking out of the side of the filter holder. If you can, you should push the plunger until no water remains. If your sample has a lot of algae or other particles in it, it may become too difficult to push the full 50 milliliters of sample through the filter. In these situations, stop pushing and note how much water you were able to filter on the datasheet. After filtering the water, separate the filter holder from the syringe and unscrew it carefully to view the filter. Make sure the filter looks intact. It may look slightly green or brown. This is the algae that chlorophyll will be measured from. Using the tweezers, remove the O-ring. And now, using a safety pin and the tweezers, fold the filter in quarters with the algae on the inside. The filter will stick a bit to the filter holder, and it requires a steady hand and some practice to do this job smoothly. Remember never to touch the filter with your fingers. Put the folded filter into a sample vial and cap it. Place a filled out sample label onto the vial lengthwise. Let's take a look at what a correctly filled out sample label looks like. Please use a fine-tipped permanent black marker to fill out the sample labels. You will fill out the lake name and ID. In addition, write CA, which stands for chlorophyll, in the parameter code box, and write MgCO3, which stands for magnesium carbonate, into the chemicals added. On the label for the second sample, write REP next to the lake name. This is short for replicate. You now have one sample completed. Congratulations! Now you get to do it all over again with the second sample bottle. When both samples are complete, wrap them completely with aluminum foil to keep all light out. This foil wrapping step is critical. Unwrapped samples cannot be accepted for laboratory analysis. Write your lake name and sample month on the foil packet. The samples go into a zip-top bag with your lake name, county, and field ID number. Fold the datasheet, place it into a separate zip-top bag so it stays dry, and place that in the bag with your samples. Storing your datasheet with the samples is a good way to make sure the paperwork never gets lost. Once you're done, store your samples in the freezer. Now rinse all filtering equipment with tap water, no soap, and allow it to air dry. On the designated turn-in dates, deliver your frozen chlorophyll samples and datasheets to the proper turn-in location. To learn more about the MiCorps Cooperative Lakes Monitoring Program, visit us at micorps.net.
Okay. So that's the video, and I hope you know you'll watch it plenty of times as if you're enrolled in this program. So a lot of details there, and it may just take some practice to get the hang of it. Just to highlight one thing from that video, if you can't filter all 50 milliliters of your sample, there is a spot on your data sheet to record that. So let's say you only could do 45 milliliters. And after that, it just got too much to push, and you're afraid that you're going to rip your filter. It's better to stop in those situations. Just write 45 down here because that's how much you were able to do, and then the lab can kind of correct for that when they do their analysis. It says sample 1 and sample 2 here. That's because every time you do one of these filtrations, you do a replicate as well. So every time you're doing 2 of these, that's why you have 2 of these that you're filling up. Okay, so when it comes to sample turn in, make copies of the data forms or take a picture of them with your phone for your records. Keep everything frozen. So you're going to put all of your filters in these little tubes, just one filter per tube, and you'll have the labels on them. They all stay in your freezer, and you have to keep them frozen. If they thaw, they're going to be worthless. So you keep them frozen. You keep them wrapped in aluminum foil, like the video showed, and they stay frozen until the moment you turn them in. So even when you're driving to your turn-in location, you should have your frozen samples in a cooler and they're on ice. So as you keep them frozen up to the minute that you turn them in, and then when you turn them in, your turn-in person should be putting them into a freezer as well. And then they actually get shipped in freezer packs to Joe. So this whole process, everything stays frozen. It'd be great if you could enter your data sheets into our database. Eric's talked about that, and Joe have talked about that already. And then you do the turn-in on that turn-in day. So here's, once again, just as a reminder, your turn-in location is on your schedule. Now, sometimes sampling goes wrong. You'll make a mistake along the way and will end up having to reject the sample. Of course, it's all just part of a learning process. Pretty soon you will all be experts at this, but there are some things that tend to happen to people, and I have them listed here. Remember to refer to your sampling schedule to remember the dates you should be sampling. So if samples are collected outside of the proper interval, then we can't trust that data going forward in a long-term data set. We always have to follow the procedures for data quality purposes. A few volunteers every year, despite our pleadings, will use that blue separator sheet as their filter. You can't do that. So remember not to do that. Your sample will get rejected if you do that. If the samples are not wrapped in foil, we have to reject the sample. Remember that we're dealing with algae here, and algae like sunlight. So the whole point of the aluminum foil is we're 100% blocking it from any kind of light that could prevent that. Otherwise, the light would still allow for a little bit of growth. So you keep stuff wrapped in foil. And then finally, incorrect delivery may ruin your samples. So if for some reason you can't follow the standard directions, which are on that particular date and time you turn your sample into the turn-in locations, you have to contact us to get alternative instructions. If you end up just shipping a sample without people knowing it, they they won't be looking for it, and it will probably thaw, and then it will be rejected. So if for whatever reason we really want you to just be able to follow the normal the drop off directions, but if something has come up in your life and you can't, please contact us before, and don't take it upon yourself to just doing what you feel like is right. It probably isn't. Contact Joe and Eric to get those instructions. Okay, I think that is it for what I have to say. And we have some little bit of time for questions, which I am happy to answer.
Okay, thank you, Paul. That was great. And there are a lot of steps to it, but like you said, once you get a little practice, it all makes sense, and hopefully you guys have a lot of fun with it. I don't have any questions currently. Please enter them in either into the chat, or if you want to unmute yourself, feel free to do that as well. We'll just wait a couple moments.
Mm-hmm.
I'm trying to think of some common things, Paul, for
Yeah, me
that
too,
can
actually.
occur.
Yeah. Welcome
I
back to the
can
scene.
comment on some things.
Oh,
Hey, all
man.
right.
Paul, you mentioned, the common reasons for rejection, and it breaks our heart whenever we have to let a volunteer know we had to toss their sample out. And it seems particularly true for chlorophyll, right? Because you go through so many
Work.
steps to collect it and filter it and drive it to the drop-off location. The last thing you want to hear is, it was for nothing, right? And so be sure to review those steps that Paul mentioned, and those common things. Of course, the filter separators and so forth. And one thing that I'm not sure we've mentioned throughout today, but maybe we have, but it bears repeating, is that instruction of when you include your paperwork with your samples, to fold that up, stick it in its own little Ziploc, little sandwich bag, and put it in with your samples, because that protects that paper from getting wet. We get a lot. I open a there's I'd say 5 to 10% maybe of that high of samples, phosphorus and chlorophyll, that I open up, and the data sheet is soaked through. It's completely soggy. You have to be very, very delicate with it to pull it out. And there's important data on that data sheet, right? And it may be the only copy if you volunteer didn't make a copy for themselves. And so I do have a clothesline with little clothespins literally in the lab that we hang them up on to dry gently before we lose that important information. So just that simple step of using a little Ziploc bag will protect the integrity of your data. Another thing I see sometimes, the labels on the sample vials, they have to go on lengthwise. Sometimes people will put them on and they overlap. And so I have to peel the label off to be able to read it, put it on longwise so you don't cover up any of the information on your label.
I see a question here. What about lakes in other counties? So it's funny because the same question came up in the in-person training. I'm like, maybe I need to do this differently. The slide that I showed of the sampling schedule was just a screenshot of a piece of paper. So all the counties are on that schedule. You just can't go off of my slide. You actually have to find the schedule, and you'll find your county there.
And I have one that was directed at me, but I think we can answer this. Are chlorophyll refill kits and data forms still on their way? And I'll answer that quick. The answer is yes. If you chose to have us mail you your refill kits, or mail the full chlorophyll kits, that is being sent here in the next handful of days, and so you should be receiving that. You'll have the data forms, the data schedule, and a procedure checklist that comes along with that. So soon, if you don't get it by May 10th, please give us a call or an email.
Just while we're sitting here, another thing that I've seen that didn't ruin the sample. The samples were fine, but it was a bit of overkill. We talk about the importance of keeping the samples on ice. when you're transporting them, keeping them cold, and when you drop them off at the drop-off location, they will put them back in the freezer and they'll ship them to me, packed in ice packs. I've actually received chlorophyll vials literally frozen in big blocks of ice. Like they filled a tray of water and put the samples in it and put the whole thing in the freezer. So I had to wait like 2 hours for this big chunk of ice to thaw and to be able to extract the samples. That was unnecessary. That was overkill. But I appreciate the person's, dedication to keeping their samples nice and cold because it did
They
need
heard
that.
the training, right?
They did. They did.
Paul, so I'm thinking of shallow lakes that are part of the program that are clear, and the speed at which to pull that up, it can be a challenge to get enough water. Can you just talk a little, I know you already mentioned it, but just talk a little bit more on that?
Yeah, if your lake is really shallow, you'll have to pull that sampler up very, very slowly. Uh, so that's because you just don't have a lot of depth that you need to pull from. So yeah, it would take a while. Once again, it's just practice and trying different things. And
What happens if they miss a month, Paul? Like they fully intended to sample every month, but they just didn't get out to get that June sample, for example.
well, if you miss 1 month, it's okay. We'll still, we process all your samples, no matter how many you end up sending. But when it comes to the end of year, looking at your data, to properly assign your lake a trophic status, like are you eutrophic, are you mesotrophic, are you ligotrophic, we do require at least 4 of the 5 samples being properly complete. So missing 1 month is okay, and that's kind of like the grace there, but you need to at least get 4.
Yep, that makes sense. That makes
And
sense.
you
And then
need
also, oh,
8 Secchi measurements. Those are the 2 numbers you got to remember. 8 Secchi measurements, 4 chlorophyll measurements to get your stuff properly calculated in your report. So it's reflective of real conditions.
yeah, so that kind of points to the importance too of, teamwork, right? And having, a backup person who can help out with sampling if in some cases, something happens, you can't get out on the water during your scheduled time, having somebody who's like, who knows how to do it, has gone through the
Mm-hmm.
training, or you've taken them with you them and trained them yourself to go out and collect that sample so you don't miss that record.
Mm-hmm.
It makes it more fun and more safe too.
Can just encourage anyone to, here's your chance to talk to the 3 of us about, because we have a few more minutes before we have a break. I guess we could start the break soon, but and then we will be back at 3:15 to do our final parameter, which is the exotic aquatic plant watch. But again,
Any
we'll just,
any question on any parameter up till now?
yeah. Well, Paul, do you want to talk a little bit? You mentioned at the beginning for chlorophyll, algae are a key component of our lake ecosystems, but they're also, seen as this negative.
Mm-hmm.
There's too much, and what is too much, those sorts of things. Do you want to just talk a little bit about that
Hmm.
or give some
A,
insight?
a little riffing off the cuff.
Yeah. Do I have too much algae?
You have too much algae. Well, let me see this question first, and we'll get
Yeah.
any comment about predictors for you. Uh, hmm. In regards to this question, any comment about predictors for cyanobacteria presence? I don't know if I'd have a good answer to that. Do either of you guys?
predictors of cyanobacteria
Yeah,
presence.
I'm trying
Interesting.
to, yeah, Phil, maybe you could clarify for us. Do you mean things that would allow us to predict that we would find cyanos, or if you find some algae, how do you know if it's cyanobacteria? Those are kind of two different questions. There are certain conditions that may encourage cyanobacteria. So
Mm-hmm.
they're also known as blue-green algae, and some of them may produce toxins and cause what are called harmful algal blooms. So they get a lot of media coverage and a lot of interest from lakefront owners. And so, highly eutrophic lakes tend to get them, but they're not limited to that. They also tend to be more abundant, in my experience, in lakes that have zebra mussels, because zebra mussels filter feed on algae in the water column, but they kind of spit the cyanobacteria out. They don't like them. And so they'll eat the other types of algae and give an advantage to the cyanobacteria. So those are a couple of conditions that may lead to higher likelihood of having cyanobacteria or blue-green algae. And I think Paul showed a graph that shows, the different algae tend to peak at different times of year, and they tend to like the warmer conditions
Mm-hmm.
more into the summer.
It's not good. The temperature doesn't control everything, but it certainly is just one of those factors. Because you can have cyanobacteria earlier in the season too. They typically will do better in lakes that are above 80 degrees Fahrenheit. Um, so that warmth is certainly a part of it. Just the higher your nutrient phosphorus levels, the more likely they are to pop up.
Yeah, and then we can have interesting conditions later in the summer where your lake is stratified and lower nitrogen conditions can allow certain cyanobacteria who can actually pull nitrogen from the atmosphere to become competitive against superior in their competition with other algae, and you can get blooms there. Joe and I were on a lake up in the UP, and fairly great watershed, low development on the shoreline, and it was a massive cyanobacteria bloom, which is pretty interesting. So there's lots of dynamics there. Cyanobacteria, a blanket, are native species that are found all over the place. And so they come and go due to different conditions based on the species presence, and they're pretty fascinating.
In
I'm gonna
regards,
I'm
oh yeah,
gonna
go
put a
ahead,
cut.
Joe.
Oh, sorry, Paul. I was just gonna say I'm
No.
gonna put a few links to some great resources about this topic into the chat as well.
And there is a great paper out there. I'll see if I can share that. I'll get that linked during the break for showing how even in low nutrient lakes you can have cyanobacteria blooms. And they talk about the different mechanisms that can bring those along too. So I think it's funny. In general, it sounds like it depends, and there's many things acting on it.
That's ecology for you, right?
Yeah, that's
Nature.
right.
It's not as predictable as we sometimes would wish it was.
And that really gets back to Eric's just previous question from a couple of minutes ago: is how much algae is too much for my lake? I think uh, this will be a question that comes up in most lake meetings with your fellow riparians that you will have. Like, is this what are we have all these numbers? Does this mean our lake is unhealthy? And that's a question that really every lake needs to answer for themselves. What are the values for your lake? Do you want your lake to be a great fishing lake? Well, if you do, you need the base of the food chain in the lake. Like, you need some amount of algae that's going to provide for your fish. And it might be that a eutrophic lake is just the lake that you want. And to some extent, the natural condition, the natural setting in which your lake lies may point it toward being a eutrophic lake no matter what you do. Now, what you don't want is anything to go hyper-eutrophic, like off the scale eutrophic. That usually implies incoming sources of phosphorus that probably should be managed, and that will also result in fish kills and dissolved oxygen problems. So a eutrophic lake, though, may be completely natural and normal for where you are. You also may have a naturally oligotrophic lake. It's just very low algae, and you may not even have not much fish in it, but it could be a beautiful boating lake. So to some extent, you get what you get based on these natural conditions, but then, of course, where people are constantly adding phosphorus to lakes as well, and that's usually what we're trying to stop. Is that slow push toward a hyper-eutrophic state. So.
Okay. Well, thank you, Paul. That was great. Thank you, Joe, for jumping for answering and building some questions.